All right. Good morning. Good morning. Good morning. I hope that our togetherness with this invertebrate enthusiasm, our shared invertebrate enthusiasm, I hope it finds you well. I hope that you enjoyed what sure felt like spring. We had a bit of a memorial, probably, for the rink in the backyard here, although next weekend's supposed to be cold, so we're leaving the plastic out and the water is down, so fingers crossed. All right, we are going to continue our voyage today within the Onychorphorans. And then we're going to delve further within the panarthropoda, which includes, as you might imagine, the arthropods. And so part of that is going to be today going over the ecdysozoans, the larger group that contains the arthropods and panarthropoda, what panarthropoda is, and some of the synapomorphies that we're going to be dealing with over the next several lectures, because it's almost like this week and next week that we're going to be here thinking about things that are chitinous and have articulated legs. So, where were we? Where have we been? Well, we were talking about the Nematomorpha and these horsehair worms and the tardigrades and the Onychorphorans. 3 different phyla in one kind of session, and 3 pretty darn incredible groups with enormous powers of parasitism and enormous powers of resilience, and then enormous powers of slime and sociality. And that's where we're leaving off with the Onychorphorans. We're going to pick up the slime and put it in an ecdysozoan package. The ecdysozoa is this large, hyper-diverse, well-supported clade of protostomes that is characterized by molting, that they have to shed their exterior, as that crayfish is doing down here, and as this dragon and damselfly, and as the blue crab is doing there, in order to get bigger. So here's where we're going today. We're going to pick up and share a bunch of Onychorphoran stories. We're going to use the Onychophora as a bridge into the rest of the ecdysozoans and the arthropods. And in doing so, this is going to sit under the large overarching umbrella in the course of these appraising and synthesizing and interpreting things about examples about how different groups eat, about consequences to placements in phylogeny, how the generalized arthropod form as we get into the specifics of the morphological adaptations of that clade, how they've been adapted upon by evolution, and thinking about molting and jointed appendages. So where we're going to pick up right now is with the Onychorphoran. Now, you've got on the left-hand side of the slide there your roadmap to our voyage through the invertebrate world, basically through to the animal world. And we're picking up our Onychophora. So this is the sister taxon to the arthropods. You can see quite a disparity in diversity. But a long evolutionary history in both groups, and together with the tardigrades, onychorphorans, and arthropods, these make up the panarthropoda. And along with the 2 other groups here, 2 other clades make up the ecdysozoa. We'll come back to that, to the ecdysozoans. Ecdysozoans, though, are a group that's characterized by molting. And certainly when you're thinking about crayfish, you're thinking about crabs, and you're thinking about eating soft crabs, maybe you're thinking about different insects kind of emerging from your lawn in July, and cicadas kind of crawling out and growing out. You think about molting. Perhaps not with things like tardigrades, and things like nematodes, and things like onychorphorans. But all of these groups also molt. With the tardigrades, we talked about the fact that the females actually strategically use their molt to lay their eggs in, to put their eggs in, so that the males tend to molt with that shed skin and save the female kind of any hassle of that whole process. So here is a video of an onychorphoran shedding that I've accelerated by 3 times, okay? And so this ecdysis, this shedding of the cuticle, is a really important part of growing up as an ecdysozoan. Now, particularly for the onychorphorans, this exterior form is much softer. You can see it kind of as this little sweater kind of dragging around behind them there, but it would still restrict growth if they didn't shed it. So here's a cross-section of the dermis, of the cuticle of the onychophoran. So composed of alpha chitin, as all of these ecdysozoans are. Then we have 3 layers. Zoomed in here, we have, thinking about them, we have the epicuticle on the top, the exocuticle underneath, and then the endocuticle. And so none of those 3 are particularly thick, nor are any of them very hard. Okay, this differentiates them from the insects. This is if you remember about those lopapod-like legs that we saw both in the tardigrades and the onychophorans, they have claws at the end, but it's a soft animal. The hydrostat is really important here. Okay, so body length is controlled, as in any kind of hydrostatic skeleton, by these antagonistic circular and longitudinal muscles that fire across this hemocoel. So if we take a rather unkind cross-section right through the middle of our onychophoran, you can see the lopapod leg out here that's got hemal channels that kind of bind around it. But we also have the terminal claw. That's going to be really important. That's coming up. And then the slime gland. And we talked about the slime glands very briefly on Friday, and we're going to expand on that today because if you know anything about onychophorans or velvet worms, probably it's the slime or the glue that you know about. And they're kind of amazing. But first of all, with these hemal channels, and then we've got circular muscle and then ventral longitudinal muscle and lateral longitudinal muscle. So this is essentially, this is an ecdysozoan that is unlike crustaceans and insects or hexapods in that it is a hydrostat, a soft skeleton. They have an open circulatory system. Okay, so it's not a closed system. Remember, we talked particularly with the mollusks, we set up this kind of continuum of like bivalves and sedentary things over here and very active predatory things like cephalopods. And we said, well, on this continuum of open to closed and more intimately connected to the muscles that are actually doing the work and more kind of diffusely connected to via sinusoids and just kind of bathing organs in circulatory fluid, arterioles at this end. This was active. This was sedentary. So these are on the sedentary end. They do have gas-like trachea, so trachea for gas exchange. So they're too big. This is not diffusion. They also have they have a nephridia, and so these classic kind of organs for excretion of metabolic byproducts and bringing in oxygen from the outside these are really. You're going to see these terms again, and we'll unpack them in more detail when we get further into our panarthropod package and talk about hexapods or insects. Ladder-like ventral nerve cord with all of those legs on the outside. That's kind of what you would expect to coordinate all of the firing of those lopapod legs. There's males and females. They have internal fertilization. They actually have quite a complicated reproductive system. So you can see here in the image, this is an onychophoran from New Zealand that is caring for newborn young. That's kind of amazing. Some females, some species have a placental structure, and most are viviparous, so that the developing embryos are retained in a uterus, and the adult gives birth not to eggs or doesn't lay eggs, it gives birth to live juveniles. Okay, so there's the hemocoel that the longitudinal and the circular muscles are firing across, and the placenta is kind of an amazing adaptation, and so to see it appear here in onychophrans is like Let's talk about slime.
To the side of its antenna are 2 modified legs called oral tubes. Connected to these oral tubes are a pair of large slime glands, so large that they account for over 10% of the velvet worm's body mass. Muscles contract around the glands, shooting a sticky slime out of its oral tubes, aiming for the beetle. The hunter may be slow, but the glue spurts out fast.
Now, in that video, with its kind of cutting-edge CGI of the onychophoran, 2 things to take away from them. One of them is something that we talked about on Friday, which is there's one measure of the importance of the slime or the glue to the onychophoran is in the investment that it has, the metabolic investment. So taking up 10 plus% of the body cavity. That's a big investment. Slime is really important. We're going to give you another example of how metabolically important it is in a few seconds when we talk about it capturing its prey. But the other is the question mark on mussels. Mussels alone it's very hard to imagine an animal that's this small propelling slime as far as it does via only muscular contraction. That's always kind of puzzled material science, not materials, but engineers and the biologists who follow these little tiny worms. So they're active nocturnally. They have these coordinated lopapod legs that are firing that allow them to kind of crawl caterpillar-like, but not caterpillar at all, like across the forest floor. And if you remember the map from Friday, we find them in these contemporarily very disparate regions in New Zealand and Australia and in South America and the tip of South Africa and a little bit in Asia. And those are regions that would have been kind of contiguous earlier in the floating of our tectonic plates in the history of our Earth. But the slime or the glue is used to subdue the prey. And there is an amazing video that I'm going to show you now from Life in the Undergrowth. And if you're watching this on the recording, hello into the future, you're going to have to follow this link and check it out yourself, because the BBC is probably going to ask me to cut this from the recording of our lecture.
This was one of the first. Fossils very like it have been found in rocks that are 540 million years old. This is a velvet worm. It too has scarcely changed since it lived in the sea, and today it's only found in wet, humid forests. It usually hunts at night, but infrared cameras can reveal it in action. Soft, stumpy legs enable it to move in total silence, and it finds its way with long, sensitive feelers. It's a master of stealth. This cricket has huge eyes, but it's difficult to see what's going on around it.
We're going to dig forward to the end times for the cricket, where the Onychorphora bumps into it with its antenna.
There.
And there.
And the cricket is trapped.
Yeah. So trapped, and then all of this slime comes shooting out and covers the cricket.
The more the cricket struggles, the more it becomes entangled.
And so, interestingly, one of the things that's then going to happen with the Onychorphora is that it will consume the cricket with kind of a circular mouth on the underside in between those modified legs, the slime papillae, but it also is going to consume again the slime, because slime is expensive to make. Now, that footage was useful to, again, biomimetic teams, combinations of engineers and biologists who were interested in how the velvet worm can expel its slime so far. And this is where the criticism or the critical thinking about the amount of muscles and how fast the muscles has to be firing in order to squeeze out that amount of slime. And so this team then began to test. And what they found out was that it's not just the muscles. The muscles are part of it, but it's that kind of open-ended end of the slime papillae. The fact that it's loose, and then when the end is not rigidly held, that helps accelerate the spread of the liquid, of the slime that's being expelled. Now, this was called passive hydrodynamic instability, or ptII, passive hydrodynamic instability. You know about passive hydrodynamic instability if you've ever turned on a hose and then had the end free, as this firefighter did, because this is the same physical property. So the amount of water that's being spread around here, that's because of not just the force of the water coming out of the pump, but also because the end of it is passive and can move around. And just as the slime papilla of the Onychorphora is. So the engineers wrote about that. To send a spray that wiggles back and forth as fast as the one in the video, the worm would need muscles that would have to contract every 20 milliseconds, and this is not what's going on. So this elastic instability, like an unattended hose, creates the glue net that allows this Onychophoran, the velvet worm, to act as this successful predator of the tropical forest floor. And I love connecting this story, that paper, and even the gif of the firefighter back to the Attenborough video, because Life in the Undergrowth was actually, it was engineers who were in grad school and physical scientists who were in grad school watching this video that saw, literally by watching the Attenborough video, they came up with the idea that this isn't just muscles. And so we need to study this. And so these two scientists got in touch with two other biologists who they knew could collect the Onychophorans, and then they set about to do this collaborative work. So an example of how evolution has co-opted a simple physical principle for a complicated behavioral response. I love that story. And I wish we could spend more time with Onychophorans. And we kind of can, because I'm going to use them to make a bridge into the ecdysozoans, into the rest of the panarthropoda. All right, Caramel reminds me, I don't see any questions emerging as of yet. Okay, complicated slide here. Start with the world. We're going to take you in the world, still retaining a little bit of Onychophora, and we're going to go to Western North America, to Yoho National Park, and to a world-famous deposit of fossils called the Burgess Shale. And we've talked a little bit about the Burgess Shale before because it's where our understanding of the Cambrian explosion came from. The Cambrian explosion, this vast proliferation of diversity, where we see, perhaps for the first time, all of the major lineages of life, all of the branches of the Dunedin phylogeny kind of make their appearance plus or minus in that. The protozoan community predates it, but our understanding of this explosion of diversity at the end of the Cambrian has to do with this particular fossil deposit being protected in a Canadian national park. It was discovered by Charles Wallacott in 1909, and it has some of the most exquisite fine body preservation of fossils that is known around the world. Since its discovery 100 years ago, there have been similar like deposits of both age and quality discovered in China, but as a measure, that bottom bullet, that about 14% of any of the genera found in the Burgess Shale and maybe 2% of the individuals would be represented in a normal fossil record. So this is working at the kind of the 80th and the 98th percentile of preservation. And that has to do with this rapid deposition. So organisms, when alive, dying, falling, being covered up very quickly, and then very, very quickly in an anoxic bed deposited and compacted and turned over millennia into stone. And the result of that is this kind of diverse understanding, both in a tape. So here in a table, we've got the phyla that have been uncovered, that Wallacott and further paleontologists, like Whittington here, described, and you can see that most of these we still see representatives today. There's a lot of sponges and cnidarians, so that's already surprising. Those are soft-bodied things that don't tend to fossilize, so this is an important record of diversification for those phyla. But then we've got these guys down here. One of the largest categories, the third largest category for the number of genera, are unknown weird wonders, groups for which we have no modern equivalent. And you can see that here in this explosion of forms, that up until the end of the Permian, that many of these groups were around and becoming more and more diverse, but most of the groups kind of have died out before the contemporary, which is where we see at the tips of these kind of broad fans that extend up there. These weird wonders include this incredible fossil on the bottom called hallucigenia. So this is an artist's interpretation in the early 1990s, late 1980s, and this is what the fossil itself looked like. The artist's interpretation is from this incredible book written by the paleontologist and science communicator Stephen Jay Gould called The Wonderful Life, The Burgess Shale and the Nature of History. And it really, if you're interested in this kind of evolutionary biology or the history of marine organisms, extant and extinct marine organisms, please, or just good reading, pick up a copy of A Wonderful Life. And it's a story. He's not acting as a scientist in this. He's acting as a storyteller, interpreting the science of others. So that's exactly right. That's exactly right. And oh my God, okay. So let me tell you, Simon Conway Morris is the scientist who described hallucigenia. And he described it because what Gould talked about was perhaps this bizarre and dreamlike appearance of the animal that perhaps as a memorial to his an age of social experimentation. So literally it was trippy. It looked like a trip. It reminded him of things he'd seen while tripping. And so that's where the name hallucigenia came from. This is Simon Conway Morris here. This is Derek Briggs, who's another eminent paleontologist, as they explore the Wallacott Quarry, so in the Burgess Shale of Yoho in the early 80s. And there's this line drawing, this crazy looking thing, this fever dream, as you're saying, and how we understand, how we certainly don't see, or we didn't think that we saw, any extant lineages that look at all like this. And this is what David Attenborough talks about. So again, if you're joining from the future, you're going to have to find this video on your own because they're probably BBC is going to get us to cut it out. But let's share it together for a second here.
This is one of the oddest of the fossils from the Burgess Shales. It seems to have 5 legs along the bottom and curious kind of lobes along the top, which presumably were some devices which help it to feed. But what kind of animal is that, with 5 walking legs and feeding lobes along the top of its back? It was such an extraordinary thought that the scientist who described it thought there was a kind of hallucination, and he called it hallucigenia. But since then, more specimens have shown that in fact this is probably the wrong way up, and that it was really like that.
I love that.
The projections at the bottom are in fact legs, and those along the top are tipped with sharp spines that were presumably defensive.
So that's amazing. That kind of literally turns everything upside down, right? Like that changes everything. And so the papers that came out originally about hallucigenia in the 70s and 80s were followed by another stream of papers with this reconsideration of what it looks like in the early 2000s and the early 2010s. So the idea originally that it had this strange pointy walking legs with kind of feeding tubes or something on the bottom, it's upside down. And so really, we're missing, and part of the rediscovery was in using novel scanning technology to be able to see into the rock, to see the preserved fossil unexposed that had hitherto lain unexposed in the rock, to see that these tubes were paired and that they were in fact legs. And so there were spines on the back, but probably defensive spines. So a soft lopapod-like leg. And this is some more contemporary author interpretations of how it moves around Eeyore-like. So lopapod-like legs with a ring of mouth in the and teeth in the mouth, and then a line of super sharp needle-like teeth inside of the throat. That begins to align it incredibly with extant diversity. The other thing that the fossil bears, and a lot of these fossils from Canada, from Yoho National Park, are in Ontario now at the Royal Ontario Museum. And what they started to do was to look at the claws, the tips of those lopopods that we see in the Onychorphora. We also learned by looking closely they were in hallucigenia. And when we started to see cross sections of those hallucigenia claws, what we saw was stacked constituent elements. Remember the analogy that I made on Friday was like bollards or pylons, the way that you stack them up at the end of needing them for a concert or a game or something. They fit one inside each other. And if you zoom in and take a look at the fossil of the Onychorphora, you can see exterior, there's a large one, and the younger, the replacements are growing inside in that form right now. This now becomes really exciting, because this formerly weird orphaned lineage, weird wonder, so strange it's like a hallucination, now seems to have an extant similarity, a similarity to an extant lineage like the Onychophorans. This is not a claw of a hallucigenia. This is a claw of a velvet worm. Amazing. So phylogenetically, what that seems to do is to adopt this formerly orphaned lineage of the Cambrian explosion to say, well, in fact, this lineage does have a modern relative, and it is the Onychophora. So the Onychophoran-like claws and the shared constituent elements in the claws, as well as the teeth that they saw inside of the mouth, so in the throat and also in the mouth itself, started to say, look, this is, we do have an extinct cousin of the hallucigenia, of that trip from millions of years ago, and it lives in tropical forests all over the world. So the teeth, let's talk about them for a second. These are these forms on the outside. So we've got hallucigenia teeth, so a ring of teeth lining the throat, toughened plates, and the throat lined with needle-like teeth. That is the same kind of form of teeth that we see with extant Onychophorans. And so the phylogenetic placement that we saw with the stacked constituent elements in the earlier phylogeny, we tend to see slightly reproduced here with Onychophorans, and being a sister group to the clade that contains hallucigenia. That's amazing, right? So this formerly orphaned group is providing not just an extant kind of group to compare, hallucigenia to, but it also suggests things about the evolution of the ecdysozoa. So these structures that have existed or that were suspected to exist in Onychophoran, the teeth itself now provide a morphological link that unifies the ecdysozoa. So it unifies the groups that previously had only been linked by DNA. But we see mouths like this in the tardigrades and in some of the other true arthropods. And that's the kind of the extent of this phylogeny. So it places it as a morphological character state that says, okay, ecdysozoans not just molting, but also this ring-like toughened plates and throat lined with needle-like teeth. So that's kind of incredible to me. We've got this lineage, this entire phylum that nowadays is restricted. It's the only phylum that's restricted to the terrestrial environment. But we now know, because of its claws and because of its teeth, that this is a lineage millions of years later, but related to this weird wonder, the hallucigenia of the Cambrian explosion 520 million years ago. So it's linked Burgess shales to the extant diversity. It's helped bind together the Onychophorans, and they just kind of blow my mind. So I'm glad that we got a chance to talk about them in some detail today. And I also wanted to share, this is a picture that was sent to me by a student of this course, crazy, 10 years ago. But this is Rebecca Smith, the summer after she took this course, when she visited the Burgess Shale in Yoho National Park, sending me back a picture, not of a hallucigenia, but just of fossils from the Burgess Shale. So I love that. Okay, Caramel's second appearance reminds me, oh, I like this. Some of you have been familiar. Yeah, it's kind of an amazing story. So I'm glad that you were familiar with hallucigenia, and I wonder if those links to the Onychophora had been made, or if those were new. Now, the ecdysozoa, we have the teeth now, we have the molting, but one contemporary analysis, some contemporary analysis have started to push the arising of these lineages a little bit earlier than the explosion, kind of taking away the analogy or questioning how explosive, how short in time was the explosion of diversity, perhaps by putting, so here's the Cambrian, and we've got all of these lineages that now have contemporary equivalents, but down here, you can see that some of, on the bars, so there's the crown group of ecdysozoa, their modern kind of analyses, these time series time tree analyses, suggest that the growth of those, the explosion, I guess that may have been lit, the wick of that bomb causing the explosion of life and the diversity may have been lit earlier than the Cambrian back into the Ediacaria. Kind of amazing. So Ecdysozoa, of course, split into those three clades. We've got the panarthropoda that have tardigrades, onychophorans, and arthropods. We've got the nematoidea, the nematomorphans, and the nematodes. We've talked about those. We've got the scelidophorans, which we've agreed that we're not going to talk about till later as just a sacrifice to time because they're not as diverse as some of the other groups. And then we've got the arthropods. And it is within the arthropods that we see the greatest diversification at a species level of life. Multiple independent colonizations from marine to terrestrial and freshwater, and famous lineages that you know a lot about, that pop culture is full of, that your stomach might be full of already. We've got crustaceans and insects and centipedes and millipedes and all of the chelicerates, including spiders and including sea spiders. And we're going to talk about those over the next 2 or 3 days. Mites, ticks, all these things. So the chapter 20, that includes all the arthropods in your text is enormous. And so the lots and lots of the characteristics appear in these kind of, in the boxes. We'll pull out some of them just to begin with. We're going to talk today largely about articulation of the legs and molting. So, but we have also to draw on some threads that we've already laid down. That includes the fact that we've got segmentation of the body and specialization of those regions. We talked about that and words for that way back with the annelids, and I'll reintroduce them again. So we have body segmentation and specialization, a high degree of cephalization. So specialization at the anterior end of the body with really complicated nervous system appendages like photosensory systems and chemosensory systems and mechanosensory systems. A highly regionalized and specialized gut, and then growth by ecdysone-mediated molting, or ecdysis. So there's my kind of short forms, and we've got some more specifics about the jointed appendages and the number of eyes. We'll pick up on these over the next kind of week and a half with some specific examples. So let's talk first of all about the exoskeleton as a hard. So this is unlike the soft body of the tardigrade that's small enough where diffusion does things, or even the soft body of the onychophoran, which still has trachea for bringing air into the body. But having a hard skeleton has implications for how you're going to exchange gas, how you're going to grow, how you're going to sense the environment. It's going to mean more weight. It might mean less flexibility, and how muscles work is generally not going to be as simple as antagonistic, longitudinal, and circular muscles any longer. So within an exoskeleton, or a body possessing this hard exoskeleton, we also see famously across the pan or the arthropods, tagmosis, heteronomy, and cephalization. Let's talk about those for a second. So segmentation, and these are some words that we're pulling forward from when we talked about the true segmented worms a couple of weeks ago. So in serial homology, segments are embryologically similar, but diverge in function some way. So and as the example here is that antenna and legs are serial homologs. They're the same thing. They're the same thing that has a different function as it grows up. And you saw or heard, if you were listening to the Onychorphora example, that the modified slime papillae, those glands, those kind of the fire hoses, those are modified legs. That's what that kind of means. They're serial homologs of each other. So when segments, like across this centipede, are similar in function, we call that homony. Heteronomy, so segments have a different function. That would be in an insect, like the head and the thorax and the abdomen. 3 different segments doing very different thing. And then heteronomy carried to an extreme, that is tagmosis. Again, with the insect example, the head and the thorax and the abdomen. And we see examples of each of these 4 processes writ large across the arthropod world. And we're going to examine over the next few days in the chelicerates, in the crustaceans, in the hexapods, different examples. So different examples where different regions of the body have become one thing in one lineage and another thing in another. So one of the other words that was in the last that characterized these groups is cephalization. So that's thinking about the anterior tagma, or the specialization of the anterior segments, being really about information processing. And so thinking about the specialization to be a predator like the mantis, or to be a social insect like the bee here, means that those anterior segments are highly specialized to picking up chemoreceptors, and chemoreceptors here, chemoreceptors and mechanoreceptors here, photoreceptors, all different, like thousands of different photoreceptors on the front end of the body, and how all of those are based on a similar body plan. And I'll talk about, with a super simplified, almost like Legos, here in a second, these simplified body plan. So the Legos that I'm referring to are, so we've got trilobites, which is a lineage we'll talk about on Wednesday, an extinct lineage, but hexapods, so insects, and myriapods, those are the centipedes and the millipedes, crustaceans and chelicerates. Crustaceans being I should have put the Tribbiani quotes around this, but so spiders and relatives, and then different bunches of arthropods here, where the segments, so the acron, the first segment, that's A, okay, and then you can see how different segments, the legs, or the names of these different segments have different roles. And so we go 1, 2, 3, 4, 5, and then the crustacea, the first two segments, A1 and A2, are really part of their antenna. They're a pair of antenna. But in the trilobites, one of them was an antenna and one of them was a leg. And in the insects, one of them can be an antenna and one of them tends to be lost. Okay, and then we have mandibles, jaws, right? Those slicing jaws of the ant or of the grasshopper. We see those here in segment 3. Segment 3 is a palp in a spider, and in a trilobite, it would have been a leg. So the changing and the turning on and off of these different segments, this brings us back to a kind of gene that we've talked about beforehand, these Hox genes. So the developmental genes that influence, that turn on and off different regions and change what's being built as the organism undergoes development. So not thinking about evolutionary time, thinking about the change in the organism as it develops its ontology. So cephalization, different regions. We'll pull apart this as we get into these different examples. We'll talk about the different regions of specialization. But let's think about those hard exoskeletons again. We'll zoom in and take a cross-section, a look at the cross-section of the exoskeleton. We've seen views like this before as we've looked at the shell of different mollusks, right? So we've got an epicuticle on the outside, a waxy thing, lipoprotein, but it's waterproof and it tends to be very, very thin. And since most of the species diversity of this group are terrestrial insects, this is an incredibly important and very thin layer on the outside of the cuticle that prevents water loss, which is one of the hardest or one of the most stressful abiotic factors that insects have to deal with. The exocuticle and the endocuticle. So the exocuticle here, most kind of close to the exterior underneath that epicuticle, is made with chitin, this alpha chitin and protein. The endocuticle is protein, and all together we would talk about them as being the procuticle. And this is where the strength and the rigidity, that when you see the shell of a larva of a cicada that has emerged, and you can it's crunchy. That's the strength and the rigidity, and the thickness and the hardness varies across different parts of the body. So this doesn't and isn't interrupted at the joints, and it isn't interrupted at the trachea. The cuticle is the trachea. The trachea is a specialized region, a very thin region of this exoskeleton. That's kind of amazing. This is a picture of Jack, our oldest son, when he was much younger, finding an emerging cicada in the backyard. And you can see, if you zoom in here, this is what it's emerged out of for however many years or months it was certainly that it was underground. And you see these white hairs. This is the adult that's kind of breathing and pumping air into its wings and waiting for its new kind of exoskeleton to harden up, so it can begin to call or respond to calls, whether it's a male or a female. But these almost kind of like lost limbs or little filamentous, like horsehair, that's the trachea. To molt, it has literally ripped out its lungs. That's kind of amazing. Let's talk about the exoskeleton. We've got a bit of a video here.
First, the mounting pressures of its growing body crack open its exoskeleton.
Little cheerleader out.
Beneath the shell, a soft, new skeleton has been developing. In less than 30 minutes of struggling and squirming, this crab breaks free of its old skeleton, a process called molting.
So 30 minutes to emerge for this particular species. And then it begins to inflate, so it can take on the extent of this new form, and it's going to take more than 2 days to harden again. Now, when we understand how the articulated legs form, this means that its movement is compromised, and certainly there's a compromise to how it's going to respond to predators and prey. So when we think about ecdysis, or the molting of cuticle, this is under control of ecdysone, which is a steroid hormone, which we'll talk about in a second. But if we were to take a look at those cross sections of insect or crustacean cuticle, we see here that we've got an mature cuticle here that has glands and the different layers, just like the diagram we just saw. But as it begins to age, kind of clockwise from 12 o'clock, we see early in a pre-molt stage the opening up of this subcuticular space down here, and the filling of molting fluid into that space that then begins to fall and to creep along and to form along this ecdysal membrane that then starts to lift up and float above. And so then the remains of the old cuticle are cast off, and we've got a new hardened cuticle and hopefully the start of another life history stage. The mineralization is particularly, it comes into play in insects, but it's particularly important for aquatic arthropods like crustaceans or early instars of insects that are living in aquatic environments. So you can see the same kind of layers, earlier diagram, but this is this line of ecdysis here, where the molting fluid can kind of split up. That leads to those lines where it can break open. I'm kind of arching my back here to show my interpretation of what that blue crab would be doing to split along that line of that ecdysal line of molting fluid to allow the body to break out of the limitations of its old form. So for a crayfish living in the Speed or the Eramosa River, this is a regular and a common occurrence. It's a major disturbance. The other thing that I want you to think about is and we'll talk about how disturbing in a second, but consider that many taxa, particularly in the marine environment, live on the exterior bodies of these. The hard bodies provides an environment. So what does molting do to the ectosymbionts that are living on top of the body when all of a sudden it's no longer a body, but it's a husk? And then think about, our younger and softer cuticles, are they more amenable to being colonized by these ectosymbionts or endosymbionts? And what happens when you're molting into and out of reproductively active states? Because the molt that you go along isn't always just bigger. Sometimes it will become, if you're molting from an reproductively inactive state to a reproductively active state, and if you're a species that has male-to-male competition, for example, like a crayfish, you're going to have one of your chelae is all of a sudden going to be like a lot bigger to deal with kind of male-to-male competition. So what I'd like you to do for a second before we're done here is tell me about, in the chat or on the screen, you can annotate the screen with the text or throw into the chat, why is molting dangerous? Come up with a whole bunch of the list, please, for why molting is dangerous. Energy costs, I think that says. Awesome. More edible. Yes. This is awesome. Okay, let me pull these up here. Easier to eat. They're squishier. No hard point for the attachments of the muscles. That's super important, Gabriel. That's good. Gives them vulnerability when they can't run away or defend. The amount of time it takes them to do that would leave them susceptible to predation. And yes, some crabs, I see that question there, Julia. Some crabs do crowd together famously during molting. Would it take more time for hermit crabs as they would have to leave their shell? That's interesting. So they're kind of doubly exposed. So I don't know the time-based answer, but in a sense, yeah, and stress, they would be more stressed for sure. Molting gone wrong. Getting stuck in the molt. That's great. That is awesome. Thank you all. Those are all super observations and hypotheses. Okay. Now, the other thing that I want you to do, and I'm going to ask you to do, is to consider what we could do as our own species if we can create a hormone like ecdysone that would be useful, that could alter when an arthropod is molting. So, we've done this. We've made a synthetic ecdysone, and we've tried to control mosquito populations using ecdysone. And what I'd like you to do is to pull out your annotation tools again and say, okay, so we've got percent survival on the y-axis. So everything's alive at the start of the experiment. Then, over a 3-week, roughly 20-day period, we've got the synthetic ecdysone application, and show me what survival would look like if this synthetic hormone was a useful deterrent to mosquito-vectored diseases. Yeah, nice. That's exactly right. I love it. Okay, right hand up, left hand up, self 5. That's kind of perfect. Before I erase your lines, I'm going to show you what the lines from the study actually look like, because they are right all inside of yours. So what we've got here is the control, and then different concentrations. And what the researchers had uncovered was that very high concentration could drop down the percent survival quite dramatically of this, so of the mosquito. So a high initial concentration of their synthetic hormone. Now, why might they do that? Or how might that work? It's essentially the bolded bullet, that if they can cause molting, inspire molting, so put this out in an aquatic environment where the mosquito larvae are living in fresh water, and cause them to molt prematurely, they'll die. They're not ready to molt. The hormone just we take control of their information system and say, oh, well, begin to molt, but the rest of the body is not ready. And so the mosquito's life is ended. And then therefore any of the diseases that it's vectoring are not vectored to us. This is potentially useful because as an insecticide, technically it's not harmful to mammals, it's not carcinogenic, and it would be useful if mosquitoes living in areas that are insecticide resistant. Okay. Yeah, Gabe, they sure can, because to me, so this is they focused, the researchers did on that by using an arthropod specific hormonal signal, it's safe for mammals, it's safe for vertebrates, it's safe for birds. Yes. However, unless it's tailored exactly to the ecdysone of a species of mosquito, you could have a catastrophic effect in the aquatic food web if all of the arthropods molted early and died. So that's a great question, and it could have really traumatic unintended effects on other arthropods. Finally today, let's talk about legs. Let's talk about the success that arthropods have from their legs, and we'll introduce this word and differentiate. So we'll talk about bodies, anterior to posterior, being segmented, and the specialization, the tagmosis of that. But the legs, which move, we're not going to call those segments. We're going to call those articulations. And they're capable of an enormous amount of independent support and movement because of the hard exoskeleton that is firing and being drawn upon by intrinsic muscles, extensor muscles and intensor muscles on the inside of the joint, and then these extrinsic muscles that link to the inside of the body. So these articulations these leg articulations have been acted upon by evolution across the diversity of arthropod lineages writ large. So some of them breathe, some of them feed, some of them only walk. So one pair per segment, yes, but they can be uniramus or biramus. So here's a uniramus leg of a crayfish, so just a walking leg, but here's some biramus ones that have a walking part and also a breathing part. And the stability of these legs is one of the major reasons behind the success of the arthropods.
One of the secrets of their success, jointed legs.
And let's look on the inside.
With their many joints, each leg has tremendous flexibility. Tendons attached to powerful muscles produce a range of motion.
So the articles, in this case, the articulation, those are the hard parts of the appendage. And then we have these muscles, the extensor and intensor muscles that weave out and across. Now remember, across those joints, that's still cuticle, right? That's still an interior. But it is in these lineages, we see extreme adaptations for flight, for walking, and for swimming. This is kind of incredible, all based or emerging from this similar design. Here's the flexed muscles on the inside of a joint that you can see still have this articulating membrane that's bendable, but is not is still part of the exoskeleton. And there's the two muscles. An enormous amount of coordination. If you think something like this myriapod in Costa Rica, that beautiful wave-like coordination of legs from anterior to posterior, but also because any one of those legs are on the ground at the same time, that there's, if we even reduce this to an insect example, there's an incredible stability.
The ease with which they move about the world is clearly a key factor in their success.
Now I'm going to zoom ahead here to some of
Inherently
the
stable
treadmill
design.
experiment. So there's as he watches, as he films these different arthropods in slow motion, what he finds is that in any one of the orientations, that there are 3 of those legs on the ground at any one time. So this tripod analogy, or the stool analogy, leads to great stability. So flexibility and stability, and we've copied this all over. So mantis or scorpion or crabs or centipedes have all birthed biomimetic informed robotic designs for independent robots that mimic the arthropod body plan. All right. This is it. This is the end of our time today. We're going to continue digging into the chelicerate, the kind of spiders and their kin, on Wednesday. We've spent a lot of time talking about linking the ecdysozoans and some of the other part within the panarthropods and how onychophorans were a big part of that. We're going to talk about chelicerates on Wednesday. In lab, you're going to be talking about mussels. This is kind of amazing. You're going to be talking about cephalopods. So that's what hopefully you'll have done beforehand. And what to do this week. Your congratulations, you're a species. The video is due on Friday, and your case study is due on Tuesday. So I'm going to stop recording in a second. I'm going to encourage you to be good to yourself and be good to those around you, and that we will then pick up our voyage through the invertebrates together next time. Take care.